Good morning, everybody. So I'd like to start by saying that this work I'm presenting this morning is a collaboration that's been ongoing nearly for 10 years with colleagues in Europe. And this picture here presents most of them, but not all of them. And I'd like to... So we're talking about non-host resistance. What is non-host? Sorry? Okay, so good. So what is non-host resistance? So non-host resistance is that holy grail. It's that resistance in a plant species that is effective to all potential isolates of a potential pathogen. I've now lost my... Can somebody move on a slide for me? Okay, so my talk this morning is divided into two parts. One is looking at a global-wide gene reprogramming in response to adapted host pathogens and non-adapted non-host pathogens. These interactions were looked at in wheat and barley to three different pathosystems: powdery mildew, leaf rust, and magnaportha, which causes blast in wheat and rice. The second part of my talk will be looking at some candidate non-host resistance genes which were identified by Patrick Pfizer at IPK in Germany to powdery mildew. And these are three genes which are receptor-like kinases, which are recorded here as Len K1, RLK7, and RLK10. Next slide, please. So starting with the global reprogramming, looking at changes in gene transcription in response to adapted and non-adapted pathogens. This was work that was started before RNA-seq became fashionable, and in this program we used the adjuvant arrays for wheat and barley. So in each interaction, we inoculated wheat with an adapted version of powdery mildew pathogen, and then we inoculated barley with the wheat powdery mildew pathogen as the non-host interaction, and vice versa. And we inoculated wheat with the barley powdery mildew version of the pathogen for the non-host interaction, and barley with the barley adapted version, and likewise for the rust and the blast interactions. So the objectives for the transcriptome study was: one, could we identify commonalities between the response to the adapted and non-adapted pathogens, commonalities between the host and the non-host interactions in the three pathosystems and in wheat and barley? Could we identify gene reprogramming that was specific to the host and the non-host interactions? Could we examine commonalities between the three pathotypes in response to the non-host interactions? And also, could we compare the non-host response in wheat and barley? So, in looking at this interaction, we inoculated wheat and barley with the adapted and non-adapted isolates of each of these three pathosystems. We looked at 4 time points, and we looked at the early stages of infection, because obviously in the non-host interaction you don't see any symptoms. So basically all 4 time points were taken between 0 and 48 hours after inoculation. So the experimental model was that following inoculation, for the powdery mildew and for the blast pathogen, because these pathogens at the early stages of infection only infect the epidermis, we inoculated and took epidermal strips. For the puccinia, for the leaf rust, we isolated whole leaf that had been inoculated. We isolated RNA, we did a hybridization against the adjuvant chip, and identified genes that were differentially expressed. So first of all, we compared the host inoculations to the mock and the non-host inoculations to the mock. And this gave us a set of what we call the general pathogen-regulated genes, genes that were induced in response to the pathogen, regardless of whether it was an adapted or a non-adapted pathogen. We then compared within this list the host interactions with the non-host interactions to find genes that were specific to the host or specific to the non-host interaction relative to the three pathosystems. We then undertook a series of blast analysis, in paranoid blast analysis, to identify on the adjuvant chips, we have an adjuvant chip for barley, an adjuvant chip for wheat, if we could identify orthologs, so genes on each chip that were basically potential functional orthologs between wheat and barley, and then looked within this list to see whether we could find genes that were differentially expressed in the non-host interactions in wheat were the same to those induced in barley. So, what did we find? So we found that, yes, there was a general response, a common response in both wheat and barley to a pathogen, whether it was adapted or not adapted, in all three pathosystems. And you can see here, there's a lot of data here, but basically there's a significant number of genes which are commonly regulated, and there are no surprises. You know, they're the general defense-related genes, cell wall-associated genes, stress genes. We then looked at, within this group of genes, what genes were specific to the host and the non-host interaction. So if you look here at the top, we have the host interaction. We have those genes which were specifically differentially expressed in wheat and barley just to the host, the adapted pathogen. And then generally, underneath, we have those genes that were specifically changing their expression in response to the non-adapted, non-host interaction. And you can see here that now the overlap between the three pathosystems is a lot less. Okay? So there's a lot of overlap between the common pathogen-induced genes, but less overlap when we start to look at when we compare the non-host to the host interactions. We then looked at trying to compare these genes which are differentially expressed in wheat and barley across the two cereal species. Looking at the adjuvant chip, we found that there were roughly 60% of the wheat genes we could find an ortholog in barley. I think that's right. And 38% on the barley chip compared to the wheat. And the difference here in these percentages is due to the fact that wheat's a hexaploid and there's 3 copies of a gene compared to barley, which is only the diploid. So when we looked at comparing the genes between wheat and barley, first of all, this is a little bit complicated. So the bars show all the genes that were differentially expressed in the general pathogen-regulated genes, those genes that were expressed in response to both the adapted and the non-adapted, in wheat and barley, for Bloomeria, Magnaporthia, and Puccinia. Okay? So the whites are the genes that were differentially expressed for which we could find no ortholog. The grey and the black are the genes that were differentially expressed where we could find an ortholog. The black is where the ortholog was both differentially expressed in the wheat and the barley. The grey is where there's an ortholog on the 2 chips, but it was only differentially expressed in one of the cereals. So you can see for this general group of differentially expressed genes, there's some commonality between what wheat and barley are doing in responding to these pathogens. When we come to look at the non-host-host comparison, we can find that proportionally, this number of commonality between the wheat and the barley is reduced. Okay? So take-home messages from this. There's a common response, okay, to pathogens, which is likely triggered by PAMPs, by the early recognition process, or by conserved fungal effectors, which trigger this general defense response in wheat and barley, regardless of the pathosystem, regardless of whether the pathogen is adapted or non-adapted. But then when we come to look at the non-host response, there's a higher degree of pathotype specificity with the non-host response, and also a higher degree of specificity when we come to compare wheat and barley, which probably is due to at some point in the separation and speciation of these two species, there's been separate evolution in the non-host and host interactions for these three pathosystems. Okay. So I'm trying to keep this is quite a complex data set. This work has been published, so if you want to know details, it's there, but I'm trying to keep this at a very simple level. Okay. So the next part of my talk comes to these candidate genes. So these were 3 genes identified in barley by Patrick, that when you silence these genes in barley, you could now find that the wheat powdery mildew was able to grow on the barley leaves in which these genes have been silenced, which these genes have been downregulated. So next slide, please. So a lot of this initial work was done using transient systems. So Patrick has developed a very nice transient bombardment system where he can take a wheat or barley leaf, he can bombard the epidermal cells either with a construct that silences the gene in the epidermal cell or a construct that expresses a gene in that epidermal cell. So we have a transient-induced silencing system and a transient overexpression system. So initially, the first gene he identified was identified by silencing that gene in barley, downregulating the expression of that gene, now made the barley leaf more susceptible to the non-adapted wheat powdery mildew. Okay? So if you look at the first of these genes, the Len K1, and this work again has been published, and on which we've done more work, and I will use this as an example. So when he took the barley Len K1 gene, overexpressed it in a wheat cultivar called Transler, okay, a transient system, what he found is that this gene now conferred host resistance to the wheat powdery mildew, which he inoculated this wheat line with. So we had transferred a barley gene, which confers non-host resistance to wheat powdery mildew in barley, to wheat, and we now confer wheat host resistance to wheat powdery mildew. He then went on to screen a series of backs, barley backs, which were selected because they had receptor-like kinase genes, because we were now targeting receptor-like kinase genes, okay? And he found 2 additional receptor-like kinases, which we just called RLK7 and 10 for the time being, which also, when you overexpress them in an epidermis of the wheat cultivar Transler, you increased resistance to the wheat powdery mildew pathogen. Okay, okay, so Patrick and his group went on to make barley lines, stable barley transgenics, where he'd introduced a silencing construct. So now we'll come back to silencing this barley gene, Len K1, the Len K1 gene, in barley, stably, and then testing these lines with wheat powdery mildew. So here you can now find that the wheat powdery mildew, which would normally not be able to infect barley, that would be your the lower picture here, could now produce microcolonies on barley. So that picture of a powdery mildew microcolony is wheat powdery mildew on barley, because this gene has been downregulated. It's not a complete infection, you get microcolonies, you don't get sporulation. But there's obviously, this gene is having some role in the non-host response. And then the other graph is just showing you the transcript levels of this Len K1 in gene in barley. So the silencing construct hasn't suppressed the gene completely. It's a partial downregulation of expression. Next slide, please. So we then went on, and my role in this grand project was the translation into wheat. So we cloned the homologue of the barley Len K1 gene from the wheat cultivar Renan. And at the time, we identified 2 alleles, 2 genes in wheat, one on 5A, the other on 5D. And we called these wheat Len K1 and wheat Len K12. Very high identity, 94% nucleotide identity to the barley gene. And in the leucine-rich repeat region, they only differed by 5 amino acids. Okay. So we then took these wheat genes and we overexpressed them in the wheat cultivar Kanzler. The first allele, 1.1, there was no effect on the phenotype. But to our surprise, the second allele, 1.2, now conferred host resistance in this overexpression system to the wheat powdery mildew. Okay, next slide. We then took the barley genes, the barley receptor-like kinase genes, and we transformed Fielder. The idea we wanted to go from the transient overexpression system to a stable overexpression system to confirm the phenotypes. This is where it came unstuck. No matter how we tried, we've produced, Marcellus is smiling at me, we've produced over 60 independent transgenic lines for all 3 of our candidate receptor-like kinases. And no matter how hard we try, we cannot obtain a stable phenotype. With the Len K1 gene, we had one single line, which we've managed to see a small increased resistance to the wheat powdery mildew, which we can take through to the T2 generation, but it's one line, okay? So science is never easy. Next slide, please. So what do we do? So we thought, well, all the transient studies were done in Kanzler. The stable transgenics is done in Fielder. So we went back and we did the transient overexpression of the barley receptor-like kinases, but this time in Fielder, and we found no phenotype. Okay? So our hypothesis is that these receptor-like kinases are forming complexes with other proteins, and that this complex is involved somehow in the primary recognition of the pathogen, of the non-adapted pathogen in barley and the adapted pathogen in wheat. Now in Kanzler, Patrick got lucky in that the other proteins in this complex are compatible with the phenotype, with the function of these genes. But in Fielder, whatever other proteins there and the alleles of those proteins, when they interact with the Len K1 from barley, they don't give a function. There's something missing, there's something required in the protein complex to see the phenotype. Okay, so take-home message. Len K1 mediates non-host resistance in barley and quantitative host resistance in wheat to wheat powdery mildew. Len K1 is potentially part of a PAMP recognition complex, with the required protein complex being compatible in Kanzler, but not in Fielder. The wheat Len K1.2 allele acts in a similar way to the barley Len K1 allele and partly prevents the adapted wheat powdery mildew pathogen from compromising those early defense responses. So I come back to my original question. Is non-host resistance a viable option for plant breeding? I think what the studies with Patrick and other people that we've been collaborating with in Europe now for some years is that there's considerable overlap between what we understand as non-host resistance, which I now put in inverted commas, because it doesn't exist as such, with PAMP-triggered immunity and with what we know of as partial host resistance, the downstream mechanisms. And I think by understanding this pathogen-mediated induction, this general response and how it's manipulated by adapted, non-adapted pathogens, we could actually learn a lot about what is basically partial host resistance. And this study has just basically scratched the surface. So I'll now switch to a slightly sadder note, as the next slide will show. Unfortunately, catastrophically, Patrick's Pfizer passed away 4 weeks ago, on the 9th of March. For those of you who knew him, he was one of the best scientists. He was an expert molecular pathologist. Brilliant, a brilliant scientist. A lovely man, a fantastic family man. He loved his children. He had adopted children. His own girls had grown up, and he went out and he found more children to give them a chance in life. He supported scientists across Europe because he was the lead. He was the one who would write the grants and get the money. And I know of 2 or 3 scientists whose programs were supported considerably just because they collaborated with Patrick. So his demise is a loss for this community. Thank you.